runaway plasmid vector. It can also be used in gene cloning procedures. So runaway plasmid vector means that sometime there is a loss of the copy number control, which we can call as runaway replication. So whenever during the course of cell division the plasmid replication control it is lost, so such a type of plasmids they may be called as runaway plasmid vectors. So they also have their utilization in the gene cloning procedures. For example, high copy number plasmids they are mainly preferred in gene cloning because it will produce more copy of the clones of the target DNA, and in turn it will express more target proteins. So target proteins will also be produced in abundance. But this abundance of protein or expression, it is not always desirable. The reason is that there are some proteins, if they are produced in abundance, they are lethal to the cells और इससे cells की death हो सकती है. तो हम यहाँ पे एक एग्जांपल कोट कर सकते हैं like OMPA protein की, which is actually a membrane protein. And this OMPA protein इसको हम कहते हैं पोरन्स. पोरन जो हैं ये ऐसी प्रोटीन्स हैं जो कि बैक्टीरियल जो मेम्ब्रेन्स हैं उसके अंदर चैनल्स बनाती हैं, एंड थ्रू दीज चैनल्स मेटाबोलाइट्स और सॉल्यूट्स दे कैन पास, एंड इफ दे आर एक्सप्रेस इन अबंडेंस it can disrupt the physiology of the cell. And there are many other examples of the proteins. If they are produced in abundance by using high copy number plasmids, then it may be lethal to the cell, cause death of the cell. That is not desirable until you get the appropriate concentration of your target protein or metabolite. So how we can solve this problem? One solution is that to clone such gene or proteins into low copy number plasmids. तो फिर इसके अपने मसाइल हैं, क्योंकि अगर लो कॉपी नंबर प्लाज्मिड में हम इनकोड करेंगे तो कंसंट्रेशन लो हो जाएगी, तो सेल डिवाइड करते हुए प्रोटीन प्रोड्यूस करते हुए ज़्यादा टाइम लेगा, which is not suitable. So then one of the solution is that to use a plasmids that are called as runaway plasmids. So genes are cloned in these plasmids that can be maintained at moderate number at 30 degrees Celsius. So the copy number will maintain at optimum. That will not disrupt the physiology of the cell. But when temperature is increased from 30 to 35 degrees Celsius, then the replication it will increase. More protein will be produced as a result of increased expression. But within first a few hours, this high expression it is not lethal to the cell. And later on, if expression keep on increasing as a result of the loss of control of the copy number that is resulted from high temperature, then the cell death may be there. So this is one of the strategy that you can control the copy number by changing the physical factor, that at low temperature the moderate copy number will be maintained like around 30 degrees Celsius, and if the temperature is increased, suppose 35 degrees Celsius, then the plasmid replication control it is lost, and it is called as a runaway plasmid. So finally we can say that with reference to plasmids we have discussed that There may be different types of natural plasmids that harbor different type of beneficial phenotypic traits, especially antibiotic resistance, that we can use as a selectable marker in the gene cloning procedures. And then with the passage of time, scientists also developed in vitro or artificially synthesized plasmids, and we quoted different type of examples like PBR322. And by using these plasmids, we can clone a DNA fragment having the size between 10 kilobase to maximum 15 kilobase pairs.